Hello students, welcome to EPG Pathshala. I am Dr. Kapendra from Kirodimmal College, University of Delhi. Today we will discuss about the module environmental factors leading to neuronal function. After studying this module, you shall be able to understand the concept of enriched environment, understand how environmental factor can influence on anatomical, electrophysiological and neurotransmitters of the neuron. You will learn about the brain plasticity, learn the influence of environment during brain development of a child and alteration of neuronal connection in adult brain. Understand the influence of enriched environment on improvement of damaged or diseased brain. It is commonly believed that animals perceive and understand the world through the senses and that epistemic connection occurs by means of transmission of information from the world into the mind through those senses. However, it has now established that brain is strong enough and has the capacity to change or modify according to environmental conditions, commonly called as plasticity. There are large number of evidences which support that brain plasticity is strongly influenced by exposure to stimulating environment. The stimulating environment shows beneficial consequences throughout the lifespan by either creating and strengthening some neuronal connections or weakening or eliminating others. The degree of modification depends upon the type of learning that takes place, with long term learning leading to more intense modification. It is also depends on the period of learning, with infants experiencing extraordinary growth of new synapses. From past 2 centuries, there have been several evidences for claiming the positive effect of environmental stimulation and enrichment on the brain function. A modern concept for neuronal plasticity in the adult brain was proposed by Hebb, who hypothesized that when one cell repeatedly excites another cell, it causes certain changes that takes place in one or both cell in such a way that one cell becomes more efficient at firing the other cell. Hebb's view was further extended, by which includes the plasticity of many definable anatomical substrates like neuritis, synapses or complete neuron that are based on changes at the molecular or biochemical levels. At an enriched environment is enriched in terms of standard laboratory housing conditions. It can be defined as a combination of complex inanimate and social stimulation. Generally, the enriched animals are kept in larger social group in the broad attractive cages, with the opportunity for more complex social interaction in the presence of a variety of stimulating objects that are regularly changed and substituted with others to stimulate explorative behavior, curiosity and attentional processes. The definition of enriched environment is based on the comparison with alternative rearing conditions, such as standard condition, in which the animals are reared in simple cages having small social groups where only food, nesting material and water are present, and the so called impecunious condition, in which the animals are reared alone in individual cages and social interactions are totally prevented. This definition implies that the relevance of single contributing factors cannot be easily isolated, but there are good reasons to assume that it is the interaction of factors that is an essential element of an enriched environment, not any single element that is hidden in the complexity, one of the important features of enrichment study is the changes in the brain that can be detected even when the adult animal is exposed to enriched environment. One of the best examples is the study of birds that were captured from the wild environment and pearls labeled with tritium labeled thymidine. Some of them were kept in an aviary and the others were returned back to their wild habitat. After some times, the birds were recaptured from the wild and the formation of new neurons in the avian equivalent of the hippocampus in these birds were compared with those birds that were kept in the aviary. The neurons that were born during the brief captivity period were survived in the birds that were recaptured after 6 weeks from the wild as compared to those birds which were kept in the aviary. So, either the wild environment is enriched or the laboratory environment is comparatively poor or stressful for the captured birds, which showed a decrease in new neurons. Therefore, the environmental changes of individual animal and the way through which it reacts to those changes can have intense and strong effect on the brain and behavior of the animal. Several studies shows that environmental change can affect the production of neurons, causes increase in glia which was attributable to oligodendrocyte and more modest increase in astrocytes in enriched animals. The enriched environment and physical work can also enhance the neuron number in the dentate gyrus. However, the mechanism of generation of new neurons might differ between above two conditions. Enriched condition affects only cell survival, not cell proliferation, whereas physical workout such as running enhanced survival and division of neurons in C57 BL/6 mice. Cell survival and proliferation might therefore be differently regulated by different environmental manipulation within a stable genetic background. Apart from the production of new neurons in the dentate gyrus of the hippocampus, both workout and environmental enrichment results in further morphological changes. Indeed, the functional changes coupled with the enrichment might be not only to increased neurogenesis, but also due to enhanced synaptogenesis, gliogenesis and angiogenesis. Several studies explain the relationship between enriched environment and their associated electrophysiological changes. When the portion of hippocampus was taken from rat kept individually or from enriched environment, it was found that excitatory postsynaptic potential, that is EPSP, slopes in the dentate gyrus were much more in the enriched rat as compared to individually housed rat. Similarly, when rodents were exposed to a new complex environment, there are enriched hippocampal field potential. There are extensive relationship between physical workout and neuronal activity. For instance, locomotion is associated with the hippocampal theta rhythm, which is the largest extracellular synchronous signal that can be recorded from mammalian brain. These theta rhythms are also strongly implicated in mnemonic function of the hippocampal and are also represent a tag for short term memory processing in the hippocampus. In addition, when running velocity of rats on wheel is increased, it causes increased discharge frequencies of interneuron and hippocampal pyramidal cells. When amplitude of EPSP and long term potentiation, that is LTP, a physiological model of some form of memory and learning, were compared in the region of hippocampus from control and running mice, EPSPs were found unchanged in both group of mice. However, amplitude of LTP was selectively enhanced in the portion of dentate gyrus in running mice over the controls. Therefore, measurable changes in the electrophysiology can occur in the same region where neurogenesis was stimulated by the process of running, which indicate that newborn cells have a functional role that is associated with memory and learning. Although the newly formed cells make a small proportion of the granule cell layer, but they have more plasticity as compared to mature cells. Indeed, the LTP of dentate gyrus remains longer in immature rats than in adult rats. The comparison of electrophysiological properties of granule cell of the dentate gyrus was done between inner young cell and outer old cell, and it was found that the putative young cell showed a lower threshold for LTP, and these remains unaffected by GABA A inhibition, which clearly indicating the enhanced plasticity of young cells. The enriched environment also causes increased release of neurotransmitters such as acetylcholine, and also shown selectively to enhance the expression of serotonin 1A receptor gene. The physical workout is also reported to change the activity of different neurotransmitter in the brain. Workout of animal influences the cholinergic parameters such as uptake of choline in hippocampus and cortex, as well as enhanced the activity of opioid system. Moreover, running or physical activity enhances the activation of monoamines such as serotonin and noradrenaline. These neurotransmitters are also involved in influencing the synaptic plasticity and learning in the adult brain, and monoamines are also suggested to function in the process of neurogenesis. Prolonged administration of antidepressants acting on dopamine and noradrenaline receptors and electroconvulsive shock enhance the number of BRDU positive cells in the rats. Moreover, grafting fetal raphe neurons excites the proliferation of granule cells, and depletion of serotonin reduced proliferation of stem cells in dentate gyrus. The word plasticity expresses the meaning of malleability and pliability. It can be defined as the capacity of neurons or neural circuit in the brain to change structurally and functionally in response to the experience. This is fundamentally property for the adaptability of behavior such as learning and memory processes, brain development and brain repair. Experience is deciphered in patterns of electrical activity within neural circuit, which drives the different forms of structural and functional plasticity through the temporally and spatially coordinated action of particular cellular and molecular factors. Plasticity of neurons involve changes in the dendritic or axonal arborization, efficacy of pre-existing synaptic contacts, formation of new synaptic contacts or eliminating existing one, and production or signaling of neurohormones or neuromodulators. Brain plasticity can be seen during development, in adult, and even during aging. The basic molecular mechanisms involved in brain plasticity remains conserved throughout the lifetime. However, magnitude of plasticity vary between the developing brain and the adult brain, which is linked to the existence of critical periods of age specific factors and processes. Synaptic plasticity is the major factor of neural plasticity. Canadian psychologist Donald Hebb postulated the existence of synaptic plasticity in response to electrical activity pattern that might underlie associative learning. He explained the importance of electrical activity pattern to induce plastic changes in efficacy of synapse. The presence of associated activity between presynaptic and postsynaptic neurons promotes the potentiation of synaptic transmission, whereas decorrelated activity promotes the depression of synaptic transmission. Have also explained the involvement of synaptic plasticity in structural and functional changes. At excitatory synapse, homosynaptic long term potentiation, that is LTP, consists of a prolonged increase in synaptic efficacy that involves structural and functional modifications of synaptic contacts, which is induced by repeated bouts presynaptic and postsynaptic activity. The first step, the activation for induction of LTP and LTD at glutamatergic synapses, is mainly dependent on the glutamate NMDARs, D-aspartate receptors. Activation, which is thought to be coincidence detectors of presynaptic and postsynaptic activity. Induction of synaptic plasticity also required activity of inhibitory inputs, because it is important in regulating the pattern of activity in the circuit as well as activation of NMDARs. During the induction of LTP, NMDARs of opening leads to entry of calcium that activate a biochemical cascade involving intracellular kinases such as AMP dependent protein kinases, extracellular signal regulated kinases, calcium calmodulin kinases type 2, the end product of which shows a rapid presynaptic and postsynaptic effect toward the expression of synaptic potentiation. It includes potentiation of AMPA receptors mediated EPSC. The passing of calcium ion will determine the polarity of the induced plasticity. Low and prolonged calcium ion transients induce LTD, whereas brief and steeper transient of calcium induce LTP. The consolidation of long term changes of synaptic efficacy includes the synthesis of new proteins which are derived from pre-existing mRNA or through activation of transcription factors such as cyclic AMP responsive element binding protein, that is CREB, by intracellular mechanism interceded by kinases signaling. These proteins allow morphological modifications at synapse. For example, modification at dendritic synapse spines causes consolidation of changes in the synaptic efficacy. In addition, neurotrophic factors like BDNF are also involved in the expression and consolidation of synaptic plasticity. The structural changes present at synaptic site also suggest that there is also a modification of extracellular matrix in the synaptic plasticity. In fact, molecules of extracellular matrix also regulate different aspects of synaptic plasticity, and proteases that act on extracellular matrix molecules are also involved in hippocampal LTP and in the plasticity of visual cortex. Several neuromodulators such as acetylcholine, norepinephrine, serotonin or dopamine, which reflect the level of emotion, arousal, attention, motivation and affection, are also involved in the induction and maintenance of synaptic plasticity. Baer and Singer showed that odiplasticity in kittens can be disrupted by using pharmacological manipulations that simultaneously causes destruction of cortical, cholinergic and noradrenergic interventions. Environmental stimulation and molecular changes efforts were done to understand the possible molecular mechanism elicited by different environmental conditions on the brain to reveal the factors that are susceptible to be manipulated to make the beneficial effects of the enriched experience. The neuromodulators such as acetylcholine, serotonin and norepinephrine have been reported to pursue learning and plasticity process in adult brain, whereas at the same time they are also strongly engaged in the regulation of brain arousal. The neurotrophic factors or neurotrophins are a group of molecules from a class of secreted proteins that are sensitive to the environmental stimuli and exerting potent functions in the nervous system, promoting neuronal development and survival includes not only the BDNF and NGF, but also neurotrophin 3 and neurotrophin 4. The neurotrophic factors are strongly concerned in regulating the structural and functional rearrangement of neurons in response to sensory stimulation during the process of brain development as well as in the adult brain. One of the effect of enriched environment is that it stimulates the brain expression for several neurotrophic factors, ensuing increased level of mRNAs for NGF as well as NT3 in the hippocampus and visual cortex that enhance protein level of NGF, BDNF and NT3 in different regions of brain. Moreover, enriched environment raises hippocampal phosphorylation of CREB that is known to regulate expression of BDNF. Continued physical workout that is done either in enriched environment or as a single component of environmental stimulation can enhance the brain uptake of physiologically relevant trophic factors like IGF-1. These factors mediates several known effects of exercise on the brain such as increased expression of BDNF and CFOS, a neural activity indicator, protective effects against the brain insult and increased neurogenesis in hippocampus. Enriched environment is also associated with chromatin modification through increased histone tail acetylation. Another tool of investigation of molecular mechanism has been provided by conditional gene knockout technique. Genetic deletion of NMDA receptor for glutamate in the CA1 subregion of the hippocampus is associated with a deficit of profound learning which can be completely rescued in enriched environment after 2 months. However, when the NR2B subunit of NMDA receptor is overexpressed in forebrain, it enhances memory and learning ability with a ceiling effect, occluding further improvement when exposure to an enriched environment. This saturation implies the existence of some overlapping mechanism between enriched environment and genetic enrichment of the NMDA receptor functions that offer an informative example of interplay among nature and nurture, whereby deletion of gene can be compensated by means of environmental stimulation, whereas occlusion of the environmental influence is achieved by genetic enhancement of memory and learning functions. The effect of enriched environment on certain conditions like epilepsy, strokes and aging appears to be beneficial, and it is found that enrichment for 3 weeks can be induced apoptosis in the hippocampus by 45% and also found to prevent the development of motor seizures when cyanuric acid is introduced into the body. It was further analyzed that pre and post operative enrichment or wheel running can help the animal in the recovery from brain lesions. The ischemia, a condition of inadequate supply of blood to the part of the body due to partial or total blockage of an artery, is also found to be prevented by voluntary wheel running. In Gerbilis housed preoperatively with the running wheels, and after that it was subjected to global cerebral ischemia for 15 to 20 minutes, it was observed that the mortality of Gerbilis was reduced from 55% to 10%. Moreover, hippocampal cell damage was also found to be attenuated. When the rats with lesion in the region of hippocampus and in the occipital cortex were kept in an enriched environment for 2 hours in a day, their performance was greatly improved on the Hepps-Williams maze, almost same with that rat that were continuously exposed to the enriched environment. Moreover, enriched condition can also facilitate recovery when it is applied after some delay. For example, rats those were undergone for middle cerebral artery ligation and were transferred to an enriched case after 15 days of surgery. These rats were tested after 7 weeks, showed significant improvement in the limb placement and postural reflexes as compared to those rats which were kept in the standard house condition. Enrichment might also exhibit some beneficial effect on genetic condition. Deficits in non-spatial memory that is associated with the disruption of NMDA receptor 1 subunit in the hippocampal region could be rescued by providing enriched condition for 3 hours per day for the period of 2 months. Moreover, in the knockout animals, the spine and synaptic density in area CA1 can be increased by providing the enriched condition. In mice having the Huntington disease transgene, the enriched condition can delay onset of behavioral deficits such as failure of motor coordination, even though neuronal symptoms like formation of the striatal inclusion bodies were not changed. Exposure of these mice to an enriched condition can enhance the process of neurogenesis and also improved learning processes. Overall, it can be concluded that enriched environment may override some genetic constraints. The multifactorial action of enriched environment on brain plasticity can also have a favorable impact on neurological disorders. One of such case is that of developmental intellectual disabilities, a group of disorder which is characterized by cognitive impairment. This disorder has affected around 2% to 3% of the population living in the industrialized world. In this module, we will focus on Rett syndrome, which is very severe disorder that offer most striking example of pleiotropy in the human population, and on Down syndrome, that is the genetic cause of mental retardation. Rett syndrome is considered as a rare genetic postnatal disorder of gray matter in the brain that predominantly affects the female population in early age of life. The female child grows normally from 6 to 18 months of age. After that, there is rapid deterioration as well as developmental stasis, which result in a complex neurological and neurobehavioral phenotype with mental retardation. It is also responsible for severe dysfunction in the motor coordination skills. The deterioration in the brain occurs because of mutation in the X-linked gene that codes for methyl CpG binding protein, which is a key regulator of RNA splicing and gene expression with a significant action of BDNF. Whereas Down syndrome is caused due to trisomy of chromosome 21. Both the syndrome are supposed to have a common etiologic mechanism, which consists of a general dysregulation of cerebral balance involving inhibitory and excitatory drive that leads to impaired synaptic plasticity in different brain parts. The above observation is based on the experiments performed on transgenic murine models. The electrophysiological analysis in mice having neuron specific expression of mutated proteins forms or conditional deletion of MECP2 exhibit reduced cell activity in the cortical and hippocampal region and reduced LTP expression in different brain regions, an effect that attribute to over inhibition. It has also been demonstrated that MECP2 knockout mice exhibit a progressive shift in cortical excitation or inhibition balance that favors inhibition by early upregulation in the expression of parvalbumin GABAergic neurons that synapse onto the cortical pyramidal neurons. In the model of Down syndrome, TS65DN mice, studies have shown that the cognitive impairment mainly occurs due to over inhibition in the circuits of temporal lobe, with excessive inhibition, which is being confirmed by the demonstration that introducing GABA A and GABA B receptors antagonists can reverse the main deficit in these mice. The analysis of human brain samples from Rett syndrome in the postmortem clearly indicate the increased density of GABA receptors, and in Down syndrome tissues there is an impaired balance among excitatory and inhibitory system. Several studies demonstrated the beneficial effect of enriched environment in TS65DN mice, an effect that turned out to be gender specific. The complex branching in the dendritic trees is also observed in the TS65DN mice. Moreover, enriched environment when combined with enhanced level of physical exercise is found to increase the process of gliogenesis and neurogenesis in hippocampal of TS65DN mice or both genders. Now, students, let us summarize what we have done in this module. Brain is considered as strong enough that can modify according to environmental conditions called as plasticity. The stimulating environment exhibit beneficial consequences by creating and strengthening some neuronal connection or eliminating others. It can elicit several plastic responses in the adult brain ranging from biochemical parameters to neurogenesis, dendritic arborization, gliogenesis and improved learning. An enriched environment is enriched in terms of standard laboratory housing condition, that is based on the comparison with alternative rearing conditions such as standard condition or impecunious conditions. The environmental changes of individual animal and the way through which it reacts to those changes can have intense and strong effects on the brain and behavior of the animal. Several studies showed that environmental changes not only increased neurogenesis, but also enhanced synaptogenesis, gliogenesis and angiogenesis. The enriched environment causes increased release of neurotransmitters such as acetylcholine and also enhances the expression of serotonin 1A receptor gene. The word plasticity can be defined as the capacity of neurons or neuronal circuit in the brain to change structurally and functionally in response to the experience. During shaping of developing brain, genetic factors are shown to interact dynamically with several environmental factors at prenatal and postnatal level, which in combination ultimately shapes the phenotype. The neuromodulators such as acetylcholine, serotonin and norepinephrine have been reported to persuade learning and plasticity process in adult brain. The effects of enriched environment on certain conditions like epilepsy, stroke and aging appears to be beneficial. The ischemia is also found to be prevented by voluntary wheel running. Enriched environment when combined with enhanced level of physical exercise is found to increase the process of gliogenesis and neurogenesis in the hippocampus of TS65DN mice, a model of Rett syndrome and Down syndrome. Thank you.